Today we'll see histology of adrenal gland. In a human, the adrenal or suprarenal glands are a pair of flattened triangular bodies located adjacent to the superior pole of each kidney. They measure approximately 5 by 3 by 1 centimeter and have a combined weight of about 10 to 15 gram. Each gland is enclosed in a thick connective tissue capsules. The glands are composed of two distinct parts, a central medulla and an outer cortex, which differ in their origin, functions, and structure. In the adult, the medulla represents about 10% of the total weight of the adrenal gland. The parenchymal cells of medulla are embryologically derived from neural crest cells. The hormones produced by secretory cells of the medulla, the chromaffin cells, influence the activity of glandular epithelium, cardiac muscles, and the smooth muscles located in a wall of blood vessels and viscera. Most of the parenchymal cells of the adrenal cortex are under partial control of the adenohypophysis and functions in regulating metabolisms and maintaining normal electrolyte balance. Now we'll see histology of adrenal cortex. The adrenal cortex is invested externally by connective tissue capsules from which septa or a tubercle extend into the cortex. Blood vessels, nerves, lymphatics, and smooth muscle fibers are present in the tubercle. The adrenal cortex is subdivided into three concentric layers, that is, a zona granulosa, a thin outer layer adjacent to the capsules. Second one is a zona fasciculata, a thick medullary layer, and the zona reticularis, a thin inner layer adjacent to the medulla. The parenchymal cells of the cortex are arranged in clusters or cones that are separated by blood capillaries and sinusoids. Morphologically, the transitions from one layer to the next is gradual. The zona granulosa consists of small columnar or pyramidal cells arranged in rounded clusters and curved columns that are continuous with the cords of cells in the zona fasciculata. In the human, the zona granulosa may be absent in areas of the cortex. The cells have intensively staining peripheral nucleus and a relatively small amount of cytoplasm containing a few lipid droplets. Small amount of basophilic materials and abundant smooth endoplasmic reticulum. The Golgi complex is present on the side of nuclei adjacent to the blood vessels. The zona fasciculata consists of large polyhedral cells arranged in parallel columns or cords, usually one or two cell thick. The columns are separated by sinusoids. The cells of zona fasciculata have a slightly staining spherical nucleus. Binucleated cells are common in this zone. Present in generally acidophilic cytoplasms are numerous lipid droplets containing neutral fats, fatty acids, cholesterol, and phospholipids. These substances represent precursors for the steroid hormones produced by the cells. The lipid is extracted during typical histological process, thus giving the cells a vacuolated or spongy appearance. Ultrastructural features associated with steroid secreting cells are observed, including abundant endoplasmic reticulum, mitochondria, and tubercular cristae. Large number of lipid droplets, a well-developed Golgi complex, and profile of granular endoplasmic reticulum. The zona reticularis consists of round cells arranged in branching and astenosine cords or columns. Compared to the cells of zona fasciculata, the cells are smaller, the nuclei of the cells are more deeply stained, and the cytoplasm contains relatively few lipid droplets. In zona reticularis, light and dark staining cells can be identified. The dark cells have more intensely staining nuclei and lipofuscins granules in their cytoplasm. The physiological significance of the two cell types is unknown. Ultrastructurally, the cells demonstrate features seen in other cortical parenchymal cells, including abundant endoplasmic reticulum. Now we'll see histology of this adrenal medulla. The central portion of adrenal gland, the medulla, is composed of endocrine parenchymal cells called as chromaffin cells, connective tissues and numerous blood vessels and nerves. The parenchymal cells are organized in short interconnecting cords surrounded by blood vessels. Chromaffin cells of the adrenal medulla are relatively large, rounded cells. Histochemically, studies have demonstrated that two cell types are present. One contains norepinephrine and other epinephrine. Ultrastructurally, the cells are characterized by the presence of numerous membrane-bound granules measuring 100 to 300 nanometer in diameter. On the basis of staining of the granules, two populations of cells can also be identified. One population of cell containing granules with very dense cores secretes norepinephrine. The other population of cells containing homogeneous and less densely staining granules secretes epinephrine. Other ultrastructural features of both cell populations include relatively abundant granular endoplasmic reticulum and a well-developed Golgi complex. From studies utilizing the technique of cell fractionations and density gradient centrifugation to isolate the secretory granules from the cells of adrenal medulla, it has been shown that the granules contain large amount of soluble protein called chromogranin in addition to the hormones. It is not clearly understood how the low molecular weight catecholamines are retained in the substance of the granules. It appears that the uptake of catecholamine by granules occurs through an active transport mechanisms involving a magnesium activated ATPase. In a limiting membrane of the granules, drugs such as respirin, which causes depletion of catecholamine from the granules, may inhibit the active transport mechanism. The content of the granules, including the hormone and chromogranin, is released by exocytosis. Now we'll see different hormones secreted by this adrenal gland. In this adrenal cortex, the cell type, that is parenchymal cells of the zona granulosa, they produce hormone mineralocorticoids, and 95% of mineralocorticoids activity in aldosterone. Parenchymal cells of the zona fasciculata and to the lesser extent of the zona reticularis, they produce glucocorticoids, that is cortisones, and 90% of glucocorticoids activities are in hydrocortisones. Parenchymal cells of zona reticularis and to the lesser extents of zona fasciculata, they produce gonadocorticoids, that is sex steroids. Now, in adrenal medulla, the chromaffin cells are there and they produce norepinephrine and epinephrine. In human, 80% of epinephrine is produced by chromaffin cells.